Hello, explorers, and welcome to Reach the World's Explorer program. For more than 25 years, Reach the World has sparked student curiosity, bringing real-world adventure, fresh perspectives, and career pathways into K-12 classrooms through virtual exchange. My name is Tim, and today is the day you've been waiting for. We're returning to Berlin to reconnect live with our amazing Fulbright U.S. Student Program researcher friend, Ellie Kreider. Ellie told us on her first Reach the World live stream that she was excited to show us her actual laboratory where she conducts her plant research, and that's exactly what we're going to do today. I'm so excited. I've been waiting for this all week. Have you read Ellie's latest virtual exchange article on the unassuming but important lab rat of plant biology, Arabidopsis thaliana? I hope I said that right. Ellie will tell me in just a minute. I hope you came prepared with questions today because after our lab tour with Ellie, we'll get to as many student questions as we can about her work. If you're brand new to this virtual exchange, please be sure to check out the links in the chat on the right side of your screen. They will take you to Reach the World's homepage for our Incredible World of Plants program, where you can find all of Ellie's articles to date, her upcoming articles, and even later today, the recording from this live event. I am so happy to welcome several amazing school groups who are joining us live today or will be watching a little bit later. But I'm going to skip shout outs today so we can get to the main show. We've got Ellie standing by live in Berlin, and she's got a lot to show us. So let's bring her onto the stage. Hello, Ellie.
Hi, everybody. Nice to see you again.
It is so nice to see you. When we talked to you last time, it was dark in Berlin, even though it's light and sunny in most places in the United States right now. What day is it? What time is it where you are right now?
It is November 13th at 4:47 p.m.
Okay, so you
are... time has hit this place pretty hard, just like it has for you guys, I'm sure.
It gets dark so early, but it seems nice and light and bright in your lab, which I know is where you're joining us today. So I'm going to turn things over to you. I want to spend the first couple of minutes today hearing what you've been up to, how your research is going. Today, we really get to dive into what you do with plants in your lab. So you are the expert in that. I'm going to turn it over to you.
Awesome. So today I've been working on an experiment that I'm working on for another person. She had a bunch of plants that she had mutated, and she wanted to see if any of them had increased resilience to stresses, particularly temperature stress. And she mutated all these lines with genes that she thought might have something to do with it. And basically I took those plants and I cut leaves off of them, and I ground the leaves up, and I extracted the DNA. Some of you might have done like strawberry DNA extraction projects or something like that. It's pretty much like that, except it's a little more sophisticated. And then I was running what we call a PCR today. So basically I checked to see if what we were looking for, because I extracted all of the DNA. So the change that we made and the change that we're looking to check, I made that bigger and bigger and bigger. I multiplied that a lot, and then so big that it's actually macroscopic, like we could see it visibly. So that's basically what I was doing today, and we'll see in a couple of hours if it worked, but I just put it in the machine.
And you told us in your article, your most recent article, about the lab rat plant that you study. Can you tell us a little bit more about this plant? It seems in the photos to be sort of unassuming, not the flashiest of plants, not a big, beautiful flower. Why is this plant so important to you?
Well, it was actually, sorry, I was just reading this really interesting article on why it actually became the lab rat of the plant science world. And before this plant, they actually used like just any plant, basically, like whatever plant you wanted to use, you could use. And a lot of times people would use crop plants because, I mean, that makes sense, right? That's what we want to research. But because Arabidopsis has a 6-week time from seed to seed, so it's like seed to seed, which means it's really quick to grow, and it is really small, and it has very small seeds, so you can grow them very like close together and grow them in small spaces, and they make a lot of seeds. They self-pollinate, so a lot of plants you may know, like like a male flower and a female flower. When a male and a female love each other very much. But Arabidopsis actually can self-pollinate, so they don't need another plant. So that makes it very easy to control the reaction. There are a lot of other reasons as well. One of my favorites is that they chose Arabidopsis long before we started doing genetic manipulation, before we started mutating plants, precisely anyways. But now we know that Arabidopsis is one of the only plants that we can easily transform because of the way that its flowers are structured. But they had no idea about that when they chose it. So I think that's just the funniest thing. Like they chose the one that was so easy to mutate, and they didn't even know it.
Amazing. I guess sometimes it just works out that way, right? I wanted, before we stand up and get a tour of your lab, which I'm very excited to do, I think many of our students will remember last time we connected with you and you told us your favorite plant or tree was the ginkgo, right? We talked about the ginkgo, and I just had to tell you, I took a walk right after that event and I stepped on something on the sidewalk and I was like, oh, what is that smell? And I looked down and it was a ginkgo tree. There was one like in my neighborhood that just like you described. So students, if you are out walking around and you step on something on the sidewalk and you're like, ooh, that seed is a little bit stinky, look at the leaves and remember what Ellie taught you, because you may have found a ginkgo. But would you take us around and show us where you work? We have seen, I think you're in your meeting room now, but what else do you do on your daily basis? We'd love a tour.
Okay. So this is the meeting room that we have our lab meetings in and put up presentations. And then here is the hallway in the lab building. And where I want to take you guys is my lab. So hopefully you can see, this is my laboratory. So you can see this box back here. That one is the PCR machine. So that's what I put my samples in today. And then over here we have, sorry, it's a little bright, but we have these two boxes here, and they're centrifuges. So they spin really fast, so we can get liquid down to the bottom. And what else?
Where is this?
These are just
are you growing plants? Sorry to interrupt you. Are you growing plants in this lab, or is this where you like take what you grow and learn about it?
Yeah, I have some. So these are Arabidopsis plants. They're very young. These ones were plated. I don't remember when I plated them. You can't read it anymore, but these are probably only a week, 2 weeks old. They were stressed pretty hard. I have some more here that were stressed hard, and they started to flower. Can't see it very well, but they started to send up those flower stalks, even though they're very young, because they're really stressed. And so they're trying to reproduce and survive. Have their generations survive. I want to also show you
there she is.
This looks like Arabidopsis, but it's actually Capsella rubella, which is a different model organism that we use in the lab. And it's called shepherd's purse. It has that common name. And we use it because it's very closely related to Arabidopsis, but it has some different characteristics that make it suitable for lab growth, stuff like that, and a little bit closer to the oilseed crops. So canola oil, soy, I think, is in there as well. Sunflowers are part of the oilseed crops.
All right. As Ellie's putting her sample away, I hope you're all thinking about questions that you have for her about her lab. I noticed something, Ellie, that you're growing plants, but I don't see any like open roof. I don't see any sunshine coming into your lab. How are you growing plants without sunshine?
So these, we are actually just growing those on the windowsill. They're not for experiments. The ones I showed you and the ones that they've already been taken out of experiments. That's why they're so stressed. But if I was in, I'd have to go 10 minutes down the walk, and we have growth chambers there that are like the pictures I showed you. So we just grow them in there, and then we carry them back and forth when we need to.
All right, very good. All right, I got to ask my question. I always allow myself one question, but now I want to bring some of our students on. While we're at your lab, if you're able to stay there, maybe they can ask some questions about the lab or your work. And I see lots of hands shooting up with our friends at 4J. Hello, friends in Pennsylvania. Great to see you today. Thank you for unmuting. Is there any brave student who would like to step up and ask Ellie their first question?
Sit back and listen. Come down here. There you go.
How long? Wait. How long have you been working at this lab in like Germany?
Going on 3 months now. I started really like at the very beginning of September.
Oh, that's very cool.
Still new to me, but I'm beginning to learn where things are.
What's the most poisonous plant that you've ever like seen?
Maybe this is a cop-out, but I've never worked with them, but I've seen some deadly nightshade plants. But I think maybe even more than that is the stinging thistle. The stinging thistle, you touch it and it stings you like a bee, and it's not very poisonous, but you definitely wouldn't get far enough to eat it because you'd be like, ow, way before that.
Great question. I love how when you said deadly nightshade, like 6 other hands went up in the background. I think there are probably a lot of questions about that. Next question, please go ahead.
Are like some of the plants that you make edible?
You don't want to eat Arabidopsis. I don't think there's anything. It's not going to be poisonous or anything, but it's just not very good for you. I do, in my previous lab, I used to grow corn and do research on corn. You can definitely eat that. But here, no. You don't want to eat the corn either because it's transgenic. It has mutations in it.
How big are the growth chambers compared to you? Like,
Okay, so we have walk-in growth chambers, and those are like, I couldn't touch the top of it. I'm 5'2", and they probably are like about as wide as this screen, a little bit longer than my wingspan. Those are the walk-ins, and then the reach-ins are probably the size of maybe this refrigerator, except a little bit thicker. And then you can maybe from here, and then you just have doors that open like this.
thank you.
Yeah.
All right, let's take one more question from Ms. Copeland's students, and then we're going to go to Oklahoma, let some other kids ask some questions, and we'll come back to you, okay? So let's take one more question.
What has there been what I mean by has there ever been like a mistake during an experiment?
Yes, every single day.
Yeah, I mean like a bad mistake.
Like a bad, like I had to start the experiment over, or bad, like or like nuclear waste type bad?
Kind of, yeah, I've just started the experiment of it.
I do. I think everybody thinks that science is like perfect, but scientists are definitely human. So, like for every day, I have to kind of pivot from mistakes that I've made. I've definitely made mistakes where I had to scrap the entire experiment and start over. Like when I add like the totally wrong reagent. Yesterday, I did the same assay, the same experiment 3 times in a row because I don't know why it failed the first time, but then the second time it failed again, and the third time I was like, okay. And I picked up the buffer that I use, and I looked at it, and I was like, this isn't what I was using. And I was like, oh, that's not good. But I've never had anything that was really bad, like that you'd have to like clear the lab or anything like that. I was in a lab once where they had some improperly stored chemicals, and those can get explosive if you, like with the right chemicals stored the right ways, they were explosive potentially. And so they had to get like bomb teams into the lab, which was fun. Yeah. It's not really a mistake, but yeah.
What a great question. We'll come back to you for Jay. I want to give Mr. Strayer's class in Norman a chance to ask some questions. Mr. Strayer's class.
Hey, you guys, give them a welcome. Yay. All right.
Do you have any brave souls who want to ask a question?
Yep, they're coming up.
Hello, my name is Micah, and I was wondering what's the easiest plant for you to grow?
Definitely Rhabdopsis. It's so easy. You just put it in the cold for 3 days, and then you put it on a plate, and it starts growing. Corn is a lot harder to grow and so is rice, because you have to like germinate it in water separately. They're particular.
Okay, thank you. Go ahead.
Hi, my name's Nathan Romine. Why is your lab so white?
White? You know what? I've asked myself that a lot. Why are laboratories white? I really don't understand it, because then you come in here in the morning and it's like so bright. But I actually have the same question. I should do some research on that.
Okay, go ahead, Toby.
I'm Toby Johnson. Why did you choose to work with plants?
Ooh, okay. I think I said it in my article, but I actually chose to work with plants because I didn't want to work with animals. And then as I started learning, yeah, right? And then as I started learning about plants, I was like, oh my gosh, these things are like really cool. And so then I was like, okay, I just want to stay with plants forever. And they became my personality.
Okay. Hi, I'm Rich Strayer, the teacher here. Hey, I was just wondering, why Berlin? Why is that the place where you're doing your research?
Yeah. So I wanted to choose somewhere in Europe because I wanted to be around as many cultures as possible. And I knew Europe had a lot of cultures in very close confines. And for me, my development as a scientist, I want to become as global as possible. So that's what I did. And then I chose Germany because I wanted a country that had an agricultural history. And also Germany was one of the most famous scientific giants in the world. Like up until the beginning of the end of World War II, Germany was it was the place to be for chemistry and also for a lot of other sciences. And last reason is because I worked with German scientists before when I was working at the Boys Tart in New York. And I really enjoyed their approach to science. They were very methodical, and I was like, whoa, this is so different from the way that I approach science. And so I was really interested to learn more.
That's great. Hey, do I have time for one more question, or are we good?
Yes, you do. No, let's do one more question, please.
I don't know if you guys have one or not. There's a group of us in here, about 30 or so. You started off, you were talking about extracting DNA. And I was just wondering, what's the goal? What's the goal of your research with that, extracting DNA from plants?
This one, I'm genotyping. So I'm just checking to make sure that the mutation we put in is actually there. because the efficiency rates for mutations are still quite low, despite the technological advances.
Okay, that makes sense to me.
Yeah, usually I work with RNA. I extract RNA, I make cDNA, and I do PCR with that. Yeah.
What's the best plant to go about it?
All right. I think that's all from us.
Well, just give me a wave if you have more questions in a little bit. I want to bring on Miss Lakes has a question. She is asking us from on behalf of a student in Vermont, how can you grow the plants in those little packets? I think they are those Petri dishes?
Yeah, they're Petri dishes. So these, we seal them. This has, I don't know if you can see that it has stuff in it. We add a growth medium to it, and this has all the nutrients that the plants need in order to survive. So it has, oh gosh, I don't know the nutrients, but I know it has carbon sources and stuff like that. And it's really awesome because we can control what's in it. So we know that there's no bacteria in it that might do symbiotic stuff or anything like that. And then we seal them up so that the bacteria can't get in around the sides. And then we just keep them sterile like that because the bacteria grow a lot faster than the plants when there's contamination.
All right, wonderful question. So Ellie, I want to get at something that someone else touched on already, and it's that you are not working in a lab in the United States, which I know you have also worked in. You're working in a lab in Germany, and you shared with us some of the differences. But science is really like a language in a way that is cross-cultural. And I wonder if you notice any major differences between working in a lab in Germany and working in a lab in the United States.
Absolutely. And I've had this conversation with a lot of my coworkers too. So in America, okay, well, we've had a little bit of maybe more arguments is the way to go about this. But I think that working in a lab in Germany is a lot better because there's much better work-life balance. So basically I came here and I was like, okay, I'm going to work 9 to 5. And my PI was like, you can work whatever hours you want to work as long as the work gets done. And this is a European thing, but it's definitely not an American thing for the most part, especially not in science. And then the other thing is that here, when you go up and you present research, in America, you would get comments like, why did you do it for 5 minutes? Why did you use that reagent? Like, why? Like, this whole experiment is BS, like, get it out of my face. But here, people are like, they assume that you did things correctly and they assume you did things with a reason. So they're asking you, like, do you think that result that was unexpected might have been because of something like this? And I feel like it's so much more productive. It's more productive discussion. I've had some people here think that it's not as rigorous or not as strenuous as the United States model, but I don't know. It's personal preference, maybe.
All right. Very interesting. We have one more question from the chat, and then it looks like we might have some more questions from 4J. If I'm right, just give me like a little wave if you're waiting there to ask some more questions. And I'm going to bring this chat in. This question from the chat real quick. This is something we touched on with the growing room. But how can a student in Ms. Lake's class is wondering, how can the plants get sunlight? Do they even need sunlight for your purpose?
They need sunlight. They definitely need sunlight because that's where they get their energy is through photosynthesis to make sugars. We have special lights. We have special lights in the growth chamber that are on timers. Actually, wait. I have a growth chamber here. Wait, we're gonna go on an adventure. I don't know if the Wi-Fi is gonna cut off, but I forgot I had a growth chamber here.
All All right, we're following Ellie through the hallways of her laboratory in search of a growth chamber. 4S, I see you in the background. We'll bring you on next for some questions. So I see you're already.
Okay. This is a different lab on the first floor. This is a reach-in growth chamber. These are two of them. So these here, hold on. I have to make sure that I'm not opening them when it's supposed to be nighttime. We have until 6. Okay. Okay, so this is what the inside of, wow, you can't see it at all. This is what the inside of the growth chamber looks like. You can see it's quite bright. Because this here, these are lights that resemble sunlight, so they have all the same wavelengths of sunlight. And then these are very tiny Arabidopsis plants. I'm actually going to use the other one because this one is hot. It's 27 degrees Celsius, and this one's a little bit cooler. And these are actually my plants that I'm growing here, and these are exactly 8 days old. Now, don't talk about my contamination. It's not good. But yeah, that's what it looks like.
Very, very cool, Ellie. Thank you for showing this. I don't know why I was imagining like a greenhouse, but that seems much more right for a lab. I'm going to bring up some friends in class 4S. Hello. Joining us from Pennsylvania as well. If any of you wonderful students are brave enough to ask Ellie a question you'd like to know about her plant research, I will encourage you to unmute, please, and come on up and ask.
Friends, let Cameron speak. Go ahead.
What made you like plants?
I think it was just, okay, because plants, they can't move, you know? Mm-hmm. So like, well, for the most part, like if something hurts us, we just run away, right? But for plants, they have to do it all through chemistry. So they have to they don't even have excretion systems. Like they can't poop or anything. They have to do it all through chemistry. And I'm like, that's fascinating. They have to, I don't know, they just make it work, you know? And somehow it seems like they're more resilient than we are to a certain extent.
Thank you. Thanks, Cameron. Okay. Remy. Come on, Remy.
How cold does the Arabidopsis plant have to be able to start growing?
How what?
How cold does the Arabidopsis plant have to be to start growing?
Exactly. It has to be at least 4 degrees Celsius, which is a little bit above freezing, maybe like 40 degrees Fahrenheit, something like that. Basically, it's just a winter. It experiences winter.
Cool. Hilda?
How big is the whole lab?
Well, it's a pretty big building. It's 3 floors, and then it's 4 floors actually. And yeah, it's pretty big. But my lab is we have the whole 4th floor all to ourselves. So a lot of it's offices, not all of it's the really cool science stuff.
Whoa, what's wrong with our space?
How many plants have you studied?
I've studied Arabidopsis, corn, capsella. I've studied rice. I've studied tomatoes. I've studied Brachypodium, which is a rice model. What else have I studied? Well, I count home experiments because then we'll be here all day. And lettuce. I've studied lettuce as well.
Oh, thank you.
Oh, and algae. I've done algae. Oh, algae's fun. People don't know. They're like, is it a plant? Is it not a plant? Nobody knows.
Do we have time for one more?
Yes, please. Let's do one last question.
What was that? What's your favorite color and what's your favorite food there?
My favorite color is green. I like really light shades of green, like something like this is nice. I don't like dark greens. And my favorite food here. Okay, there's this food here. It's called Greece, which is not Greece the way we think of Greece. It's GRIES, and it's kind of like tapioca pudding. If you guys have ever had tapioca pudding, or like a porridge of some kind, and it's made from semolina, which is a plant that we don't really grow as much in the United States, and it tastes really good. It's just like a milky pudding, and then you put like fruit stuff in it.
All right.
Thank you.
Excellent question. I'll move you backstage for us. Thank you for the questions. We'll come back to you if we have some time for some more questions. Ellie, I had a quick question before I go back to students about, you shared in your article a picture of your lab notebook. And notebooks are very familiar to many of us in school environments. But in my experience, I see a lot of people putting things on computers these days. And I wonder, why do scientists like yourself continue to write things by hand in notebooks the way you do? Was there a reason that you use a paper lab notebook instead of taking notes on like a computer?
Absolutely. Two reasons. One is that computers are easier to hack than lab notebooks, and we need to protect our data from other scientists sometimes because they'll steal publications. And two, because it's the law here in Germany, the Research Institute has to have paper copies of the research that's being done. Okay. I was in a lab once that you could choose, but a lot of us chose paper anyway because it's just easier. And you don't really want like chemicals on your computer either. That's not ideal.
That was my guess, that you were going to have things on your fingers that you would get your keyboard all sticky, or the way we do when
I knew people who because you see these keyboard. I knew people who when they went to work, they put a different keyboard cover on their keyboard.
Okay, very interesting. I'm going to loop in two questions from Miss Lake's class before we go back over to our friends at 4J. So 4J, you're on deck. Miss Lake students would like to know, what is the coolest tool in your lab? That can be your personal opinion, or maybe everybody agrees the coolest tool in the lab.
I think that it's probably unanimous that the fluorescence microscopes are pretty much the coolest machine. You saw pictures. I sent pictures with the fluorescent Yes, okay.
Yes, yeah.
So obviously it looks freaking cool, right? I mean, come on. It's not fun to do though, because I spend 5 hours in front of it every time I use it, practically. Selecting seats.
Very, very cool. Excellent question from Miss Lake students. And this is a very interesting take on this as well. Do plants have something that is like a brain? You expect me to answer this in a minute? That's crazy. Okay, the long answer is the shorter answer. The interesting answer is that there have been associations called the Plant the Plant Neuroscience Association. These do exist because they have nervous systems, not just exactly like us, but kind of like us. And they have memory, and they have purpose, and they have a lot of individualism kind of as well. And so people are yeah, sure. But because the idea is that because science is so mammal-centered, the answer is, I can tell you, is no. They had to change their name of that association, and we're taught no. But a lot of plant scientists that I know think maybe a little bit different. Very interesting. I feel like there's another hour-long live stream on that topic. The students are asking the hard questions today. That's a Very, very good one. Whoever asked that question in Miss Lake's class, you're onto something. Keep investigating, and there's more to that story. All right, 4J, welcome back to the camera. I can see that you are ready with some questions. I'm going to turn it over to you.
And Ellie, we could probably ask questions for another 45 minutes, so just tell us when to stop. Alrighty.
Okay, so how does the DNA extraction process work? Like, how do you take the DNA out?
You grind it up, so you basically break all the cells, and then you use a chemical. It's called isopropanol, which you might actually have in a lot of your bathrooms, and that lyses the cells some more, and then it also extracts it causes the DNA, which is soluble in water. So the DNA is kind of dissolved in the water, and then you put isopropanol on and it falls out of solution. So it starts getting really clumpy. And then you hit it with, and then you take everything, you pellet that down, you put it in the centrifuge and it goes down to the bottom. And then you take everything else out, and then you you make it go back into solution with a different buffer that it can dissolve in. Okay. It's not super pure. This method is what you call a dirty method. It's not super pure, but it is good enough for just checking, basically, if the mutation is there.
Okay.
Yeah.
Why is the corn in your lab not edible?
Because we changed the genetics of it. So if it hasn't been cleared for human consumption, we're not allowed. This is my big question too, because when I was we were growing corn in a growth plot back at my home college, and I was like, can we have a barbecue? We can have normal corn. And my lab professor's like, no. She's like, we can't. She's like, you might eat it by accident, and you don't know what the unintended things are. We end up made it poisonous or something.
Okay.
Why do you have to put the things in the growth light thing if there's sunlight outside?
Because sometimes it's winter, like now, and we want the plants to think it's summer. And sometimes we have droughts, or we have, cold snaps, and we need to control the water and the temperature. And sometimes we want it to be day. Sometimes we want to study the night cycle, what do they do in the night? And we don't want to come here at 3 a.m, so we just artificially make them think it's nighttime earlier in the day. There's a lot of reasons for it.
What are the mutations that you're doing on it?
I'm looking at these genes that we think may have something to do with the cold stress response. So when plants are stressed with cold, they change their genetics so that they can adapt to it. But we don't actually know really what they turn on and what causes the changes. So I'm working on that. I have a couple of candidate genes, and basically what I do is I knock them out. So I say no more gene in the plant, and then I see if that affects the stress response. And if it does, then maybe it has something to do with it. That's kind of a lot of science, although it sounds a little bit tedious.
Thanks.
What a great question. I think it sounds fun. You get to just guess which gene you want to try, and then you grow the plant and you see if it worked. And if it didn't, you try another one. Yeah. Like a ton of fun. Great questions from 4J. Do we have any other questions?
Yes. Do we have time for one more?
Yeah, let's do one or two more.
Marcus, did you have one too?
Why didn't you want to study animals?
Because when you work with animals, usually you have to kill them afterward. And I know, and they do it in a very humane way, or so they tell me, and I know that it's true, but it is still hard, and I didn't think I could do it.
Thank you.
Yeah.
Okay, we do have one more, if that's okay.
I think you've, Ellie, just created a lot of future plans.
I know, that was, did you see their faces?
Based on the,
it's not that bad. If you're studying insects, they just throw some anesthesia on there.
I think Marcus's question will have a happier answer.
Have you ever played Grow a Garden?
Okay, wait, listen. Stop and listen. Can you explain what it is?
Have you ever played Grow a Garden? It's like a game on Roblox where you can grow a garden and stuff.
I don't think I'm part of that generation. I'm sorry. I actually don't even play mobile games. I'm bad.
Say thank you.
Thank you.
Thank you.
All right. Thank you, 4J. Thank you so much for those great questions. I'll move you backstage. 4S, I see you. Do you have any final questions that you would like to ask Ellie before we sign off?
You're okay.
So, what was your hardest or plant that took longest to grow?
What was my hardest what?
Plant that took longest to grow.
Corn. Corn takes the entire summer to grow. No, it's so easy. When you work with algae, you can start working with them within 48 hours. When I work with Arabidopsis, yeah, I know, it's disgustingly good. And when I work with Arabidopsis, I have to wait 14 days before I can do any of my assays. I know, but corn? It takes 2 weeks to even get up this high. No less do experiments with it. It was disgusting. There's a reason that we use Arabidopsis. How about that?
Very good question. I've watched corn grow very, very slowly over the course of a summer. And have you ever heard the saying knee high by the 4th of July? It's like a measurement saying about when corn is growing, at least in the part of the world where I grew up. I think Arabidopsis grows a lot faster than that. All right, for us, any other questions? I see one, couple hands.
Go, Thomas.
Is cotton candy grapes genetically modified?
Well, yes. The answer is yes, because all grapes are genetically modified, okay? Don't even talk to me about seedless or not seedless. You think it was like that normally? No, that was selective breeding. But I don't know about that particular cultivar, if it was GMO or not. If it was some human went in there and did something. I don't know.
So, Ellie, are you saying that anytime we eat a grape that doesn't have seeds in the middle, that it has been genetically modified by a human to not have seeds in the middle?
Is that so surprising? Yes, that's what I'm saying. Because how would it grow like that in the wild if it has no seeds? It can't reproduce.
It's a good question. Some of our friends in Pennsylvania might have grapes in their lunch today, and you had never thought that. You don't say, ugh, I don't want seeds in my teeth while I'm eating my grapes. Well, a scientist like Ellie at some point said, wouldn't it be nice to have grapes without seeds that would not get in our mouth while we ate them? And voila, you have seedless grapes.
Thank you.
We have one more?
Yes, please.
Okay, Kyle.
What's the biggest plant in the lab?
In the lab? Capsella is
Your lab.
It's a little bit bigger than Arabidopsis, so I'll say Capsella. Yeah, but they're both pretty small. They're this big.
All right. Students, you had such great questions today, and I'm sorry we can't get to all questions, but I know you've got to go on with your day. I'm going to bring 4J onto the screen so you can see each other. And you can all say thank you to Ellie. Thank you. The teachers are so well trained at Zoom etiquette. I've immediately muted everybody, but it's okay if you want to unmute and say a big thank you to Ellie for answering all our questions today. You guys are the best. Thank you so much for joining today. Ellie, before we go, I have one just fun question for you. I assume that everybody you work with likes plants. And finds plants interesting. Does the lab grow any just beautiful ornamental plants to add some character, do some air cleaning, just have some plants in your environment the way many of us do in our homes?
Absolutely. You saw the spider plant that was next to my windowsill in the meeting room. We also have a cactus in there. We have a succulent in there. And we celebrated our 100th anniversary a week or two ago. And they grew herbs and gave us herbs to take home. So now I have like 3 herb plants. Yeah, we just do it for fun, for sure.
That seems like a super appropriate gift to give people who work in a plant lab. Wonderful. Well, the time always flies when we're talking to Ellie, and unfortunately, we've got to wrap it up here and send our students on to lunch or wherever they are in their day. Hopefully, you have a greater appreciation for your seedless grapes after that wonderful lab tour and talk from Ellie. Thank you so much for sharing your cool work and passion for plant research with us once again, Ellie. We're going to reconnect with you again in a few weeks for our final live stream on this virtual exchange. But in the meantime, our students have said goodbye and may already be off. So I'm going to say auf Wiedersehen on their part. And for anyone who's watching the recording, you can say a little German goodbye as well. Say auf Wiedersehen. Teachers, please keep checking the Incredible World of Plants Journey homepage for more articles. There's more to go. We have more to discover. We're going to wrap it up in the next few weeks, though. And until next time, students, keep exploring, stay curious about plants, go step on a ginkgo seed on the sidewalk and see what I'm talking about, and keep an eye on the other green things in your backyard. Thank you so much for going on this Reach Your World journey with us. Tschüss, auf Wiedersehen, and goodbye for now. Thanks, Ellie.